Welcome back, class. And in the last section of the unit, I'd like to talk about how material moves across a selectively permeable membrane. But before we talk about that, I want to review a few terms. First, I want to review what a solution is. Remember, a solution is a well-mixed mixture. And there are two parts to a solution, the solute and the solvent. Remember, the solvent, like example water, is the material that's present in the greater amount and is what you're dissolving into. The solute is what you're actually putting in, and that's getting dissolved. Concentration is a measure or ratio between the solute and the solvent. The more solute you have compared to the solvent, the more concentrated the solution is. Today I want to talk about some of the different forms of transportation or transport. And what am I talking about by transport? I mean the movement of material across the selectively permeable membrane. First, we'll talk about the types of passive transport, which are types of transport that do not require any energy to get them to occur. The first type we'll talk about is diffusion. Diffusion is the movement of particles from an area of high concentration to an area of low concentration. And this occurs because of the motion that occurs with molecules. Molecules have kinetic energy. They're moving around. And as they're moving around, they're bumping into each other, colliding, ricocheting, and moving into places where there's less of them. This doesn't require any energy on the part of the cell to move the material across the membrane. It's a naturally occurring process. So you can see in this image here, the area with the higher concentration is on the left-hand side, the one I just checked right now, little arrow, whereas the area with the lower concentration is on the right side. So which way would the material move? The material would move this way, from an area of high concentration to an area of low concentration. There are less particles on the right side of the membrane than the left. So the particles will continue to move into that direction until they balance out. That balancing is called equilibrium, or net equilibrium. That's when the concentrations of materials are equal on both sides of the membrane. That doesn't mean the particles stop moving. They still have energy of motion. They still have kinetic energy. They're going to continue to move in both directions, but it's going to be kind of a give and take. Pretty much an equal movement. Some molecules across, some back across the other side. Here's an example of the movement of material over time. Notice you have a high concentration of material here. As time passes, notice the particles start to move through the phospholipid bilayer. And as time passes, you'll notice that the amount of particles begin to balance out, so that eventually you have net equilibrium. The same concentration of solutes on both sides of the membrane. What about when we're talking about water? Water has its own special term. Water does diffuse like other substances, but when we're talking about water, we're talking about osmosis. Osmosis specifically is the diffusion of water through a selectively permeable membrane. Water can pass through just about any membrane, whereas sometimes the solutes that are dissolved in it cannot. So sometimes to balance out the sides of the membrane to create equal concentrations and equilibrium, it will be the water that will have to move to balance out the solutes. Now, when we're talking about osmosis, there's three terms we have to be familiar with. Starting first with the term tonicity. What is tonicity? Tonicity is the measure of the amount of a substance, like a solute, dissolved in a liquid. Most of the time, it's water. There are three different forms of tonicity that can occur on a long membrane. And these are really relationships, one side versus the other side. You have hypertonic solutions, in which there are more solutes on one side of the membrane than the other. You have hypotonic solutions. That would be the other side of the relationship. If you have a hypertonic side, that means there's going to be another side that has less concentration of solutes. That would be the hypotonic side. And sometimes you'll have equal concentrations on both sides of the membrane. That's called an isotonic relationship. Water will always move towards the hypertonic side. That's called osmotic pressure. Because the side that's hypotonic has less solute particles, which means it's going to have more water in relation to the other side. So therefore the water is going to have to move from that side to the other side. But remember, tonicity, when we're talking about tonicity, we're talking about the measure of the solute. So when you're trying to identify which side is which, remember the terms hypertonic and hypotonic refer to the amount of solute. Which side has more solute? That's the hypertonic side. Which side has less solute? That's the hypotonic side. So let's look at this example here. And I can't show you the whole thing because it's a PDF, but you can see on the left-hand side, we have a lot of these sugar molecules, which you can see in red here. There's a high concentration of those sugar molecules. But on the right side, you'll see there's only a few sugar molecules. Therefore, the side that is hypertonic is which? If you said the concentrated sugar solution, this one here, you are right. That's the hypertonic side. Whereas the diluted sugar solution on the right-hand side is the hypotonic side. So which way will the water move? Okay. The water will move to the side where there's a more concentrated solute. So the water will actually move, as you can see in the direction of the arrows, it will move this way, to the side where there's more solute. So that could actually create a scenario later where there's actually, and I apologize for this crude drawing, something that might look like this. With the membrane here, there might actually be more water here and less water here. Why would that happen? If they're isotonic and equilibrium has occurred in their balance, shouldn't they be the same? The problem is there's more solute on one side than the other. So it requires enough water to create a balance between the concentrations. So to create a balance from both sides, you might need more water on this side than this side because of the higher concentration of solutes. So even though the water levels are not the same, the concentrations of solute to solvent is the same on both sides. And so now they've reached equilibrium. Here's another example. Okay, which would be the hypertonic side and which would be the hypotonic side? The little black dots represent the solutes. If you said the inside was the hypertonic side, you are correct. Whereas the outside is the hypotonic side. So which way will the water move? The water will move into, we'll call this a cell here, it will move into the cell, forcing the cell to swell. Now, how do cells cope? Well, cells have things like contractile vacuoles, which we'll look at later when we talk about protists, but these are structures that can regulate the amount of water in the cell. Multicellular organisms can work to maintain homeostasis by affecting the concentrations of water in the bloodstream through different hormones. You have excretory organs like the kidneys, which can get rid of excess like toxic liquid waste. Plants use their central vacuoles to affect the amount of water in the cell, which can also create the pressure that gives you the swollen look of leaves that you see on a healthy plant. And here's the chart that was missing from your presentation. This is a great one to look over for the test, where you can see each type of solution and what they would do to cells. Make sure you look that over. Now, everything we've talked about so far has been examples of passive transport. Diffusion and osmosis are all examples of passive transport. So is facilitated diffusion. Facilitated diffusion is exactly what it says. It's facilitating or assisting diffusion. So it's a naturally occurring process, and things are moving from high to low still, or what we say down the gradient. And the problem is that sometimes particles that may be in a higher concentration and want to move across the membrane can't. They don't fit for whatever reason. They can't get through the phospholipid bilayer. So mixed in the phospholipid bilayer are special structures called protein channels. These are some of the things that make up the beautiful fluid mosaic model that we talked about the other day. These protein channels are almost like a lock and key kind of thing. They will accommodate certain solute particles which will be able to get through the protein channel and then move into the cell. Here's another example. You can see these solute particles here. They fit perfectly into that piece, and then they can slide through the channel and into the cell itself. This doesn't require energy because you can see there's a large concentration of particles on the outside. So they're moving by diffusion. They're just getting a little help, because otherwise these particles would bounce right off the cell membrane. They wouldn't fit in there. Now, active transport is a little bit different. Active transport is when energy is required. And energy is required because in active transport, sometimes you have molecules moving across the gradient or against it. And so what's happening is you have material moving from low to high rather than the other way around. So that requires energy. That requires, again, some special types of pumps, or some special types of proteins we call protein pumps. These guys actually push the material across or against the gradient. And to do that, they use a special molecule called ATP. ATP is the molecule that provides energy at the cellular level. And you can see here, this is where the concentration of material is higher on this side. So you would probably think instinctually the material would want to move into the cell this way. But this material is also found on the inside of the cell, and it's actually moving out. So the only way it can move out is through these protein pumps, and because it's pushing against the gradient. It doesn't want to move that way. To do that, you'll see the ATP actually provides a little bit of energy, releasing one of the phosphates, breaking the bond between the second and the third phosphate. We're going to talk about that in a later unit. Giving that little bit of energy the protein pump needs to push against the gradient. Some other examples of active transport include some things we actually talked about a little bit earlier in the unit, endocytosis and exocytosis. Endocytosis, remember, which means come into the cell, is when a cell engulfs a particle. And there's two different examples. There's phagocytosis, which means the cell is eating a particle. Like here, this is phagocytosis right here. You can see it's engulfing this particle here, forming a vacuole around it and bringing it into the cell. That requires energy. Sometimes the cell brings in liquid material. It's called penocytosis. Forms a vesicle around the liquid material, brings it into the cell. Sometimes the cell actually wants to get rid of stuff. We call that exocytosis, which means like out of the cell. So it's actually pushing the material out of the cell and into the extracellular space outside of the cell. Don't forget when you're studying to look over the section 7.3 and section 7.4 readings. Look over this presentation here for section 7.3, the recording and your notes. Also look over any practice sheets we did, like the ones on osmosis, diffusion, and tonicity. And don't forget to bring a pencil. Thank you.